All right, we are on to activity 2, where we're going to talk about chytrids and zygomycetes. So just a reminder, you're going to watch this lecture. There's some bonus videos that are embedded in the PowerPoint slides, but also I'll post separately. And then this will correspond to questions 3 and 4 of your worksheet. All right, so the first group we're going to talk about is chytromycetes. So these are the most closely related ancestral fungal form, and they are most likely having diverged from a protist ancestor. They're pretty simple and water dependent. They are unicellular, but they have a wide variety of physical forms and features. They are usually aquatic and motile with flagellated spores, so their spores have a flagella, and many of them are active decomposers, just like most fungi. So this is a really cool picture here at the bottom of a chytrid cell actually decomposing a pollen grain, and you can recognize what type of pollen grain that one is, right? The Mickey Mouse head. All right, so chytromycetes are saprobic parasites, which means that they break down they use extracellular digestion to break down organic matter. And so they usually decompose really tough things like pollen, cellulose, chitin, and keratin. They're sometimes some of the only groups of organisms that can sometimes break down these really complex molecules. There is one really important chytrid species. It's Bactritidium dendromatitis. This organism is believed to be partially, if not fully, responsible for major declines in amphibian populations worldwide. I'm sure you've heard about them, maybe. So the chytrid infects the amphibian skin, and it breaks down the keratin on the skin, and it causes spores and excess skin production, which is a big issue for amphibians since they breathe through their skin. And so it inhibits that cutaneous breathing, which is breathing through your skin. There are ways that you can test for chytrid fungi on amphibian skins, but it's a really big issue. A lot of people that do field work with different amphibians have to be really, really careful that they really well clean their boots and wear different gloves when they're going to different field sites so they don't spread the chytrid with them. And there is a video that you can watch about it. All right, so the next group we're going to move on to is zygomycetes. So zygomycetes are bread molds that do not have a fruiting body. So that means they are just vegetative body, no fruiting body, so no sexual reproduction. There are over 1,000 species, and they are typically found in terrestrial environments, living in soil or dead or decaying plant matter. We know them best for growing on old bread. So if you ever have had a loaf of bread, or feel like I see it on strawberries a lot. Maybe I'm just bad at eating strawberries. But that white fuzzy stuff, that's a zygomycete. It's bread mold, and they reproduce sexually and asexually. I think I said a little bit ago that they don't produce sexually. I was wrong. They have no fruiting bodies, but they still produce sexually, so wipe that from your brain. All right, their hyphae are aseptate, so they do not have cell walls in their hyphae. And the hyphae usually grow unnoticed in their substrate until their reproductive structures are produced. So pretty much if you see mold on your bread, it's already been there for a while, right? It only produces the reproductive structures when it's really happy, and it usually has enough sugar and it's able to really get to town. So sometimes if you like cut off a chunk of bread that's extra green, there's still other mold in it, but you're probably fine because we all eat a lot of mold in our lives, so you'll be okay. But just know that there is mold all the time. All right, so we're going to go over the life cycle of sexual reproduction in zygomycetes. So it starts out with the hyphae. So there's cell signaling that will occur within the 1N hyphae to produce within the different mating structures, so like plus and minus. The gametangia produced, which is this kind of like blob structure that comes off of the hyphae. Then the two mating types will fuse together and fuse their cytoplasm through plasmogamy to form a zygosporangium, and the zygosporangium is N plus N. It's this little Oreo looking thing in the middle. And then the zygosporangium will go through mitosis and break up a little bit more, and then we'll see it's still an N plus N here, go through mitosis, and then the nuclei of the zygosporangium will then unite via karyogamy to form the zygote. So then it's 2N zygote, then it goes through meiosis really quickly to form the zygospore with a tough outer wall on the 1N nuclei, and under favorable conditions it will produce zygospores that are 1N, and that will mature into conidia that will spread the spores throughout. So just to review that a little bit, I feel like there was a lot going on. So we have these different mating types that reach out to each other to form the gametangia. You can see them reaching out to form the gametangia right here. Once the gametangia, which is one end, is meeting, we have plasmogamy going on. So though that becomes the zygosporangium, which is N plus N. It grows via mitosis. Eventually it will fuse together via karyogamy to form the 2N zygote. The 2N zygote will then go through meiosis to divide from 2N to 1N, usually pretty quickly, to form these zygospores, which are 1N. So go through that a couple times and try and draw it out because it's confusing. All right, and then asexual reproduction. The 1N hyphae in zygomycetes can also reproduce asexually. In this case, it just goes through mitosis and forms 1N spores. This creates obviously less genetic diversity than you would have in sexual reproduction, but it does spread pretty quickly. All right, so when it undergoes through asexual reproduction, the 1N spores will spread through the air. The spore-bearing structures kind of look like lollipops, I think, which is going to be different from when we see different asexual structures in ascomycetes later that are going to look more like trees. So here we have a sporangiophore with the sporangium on top that spreads the spores, and that usually branches directly off of the hyphae. And those are you'll see it called conidia sometimes too. So sometimes it's called sporangiophore, but the conidia is the head part that's on it. Okay, so just take a closer look at the sporangia. So like I was saying earlier, the sporangiophores are the stalk, and the sporangium is the head part on the top. And it appears to, it looks like a lollipop. Again, you're seeing the circular sporangium shape on the stalk. And then eventually the sporangium will open up and the spores will be spread. Okay, and this is just a review of the life cycle, so we can talk through it one more time. Let's start off in the mycelium with two different mating types. There's plus and minus. They reach out to each other to form the gametangia, which is this Oreo-looking thing in the middle that are still having haploid nuclei. They're still separate cytoplasms, but through plasmogamy, they fuse together to have the dikaryotic nuclei, which is N plus N. That'll mature and form the zygosporangia, which is heterokaryotic or dikaryotic, so it's N plus N. Then via karyogamy, the N plus N will come together to form a 2N embryo, and then the diploid nuclei will go through meiosis to create spores that'll spread off of the sporangium. And remember, in zygosporangium, they're called zygospores, or in zygomycetes, rather, there are zygospores that are contained within the zygosporangium. All right, that'll be all for that one.